Hello and welcome to chapter 6.1. Okay, genetic control. So we learned a little bit about DNA in the last chapter, and you learned quite a bit about it in STEM. So we're going to go a little deeper in chapter 6. DNA carries instructions for the construction and the behavior of cells. And the copies, so DNA copies itself in order to do this. And the nucleus must pass on the exact copy of genetic material to the daughter cell in order for there to be exact instructions for the construction and the behavior of the cell. Okay, DNA stands for deoxyribonucleic acid, and then its close cousin is ribonucleic acid, which is RNA. These are polymers, and they're polymers of nucleotides, aka polynucleotides. Nucleotides are made up of a nitrogen-containing base, a pentose sugar, and a phosphate group. And the three of those things together are called a nucleotide. So it's really important to remember that. A lot of students think that the nitrogen base, the A, T, C, or G, is what a nucleotide is, and that is incorrect. It's the combination of the nitrogen, the 5-carbon sugar, and the phosphate group. Okay, so the nitrogen-containing bases, the nitrogenous base, are adenine, thymine, which is in DNA only, guanine, cytosine, and uracil, which is in RNA only. Okay, alternating sugars and phosphates are linked together with bases projecting sideways. Right? So the DNA molecules are made from 2 polynucleotide strands lying parallel to each other in opposite directions, and strands are held together by hydrogen bonds between the bases. The purine bases are adenine and guanine. Okay, and you can remember that. One way to remember that is purine bases are pure as gold. Okay, so purine bases are adenine and guanine. And the pyrimidine bases are cytosine and thymine. And the complementary base pairs, A always pairs with T in DNA and U in RNA, and G always pairs with C. Okay, so here's some pictures of that here. So you see the nucleotides are going to be the phosphate, the sugar, and the nitrogenous base. And those 3 nucleotides make up the entire one whole side in the middle of the sort of rung of the ladder. Right? And then the two sides are held together through hydrogen bonds. So DNA is referred to as a double helix, and it is called right-handed. And normal, standard, typical DNA is right-handed. And if you see this image on the left here, this person has their right hand doing a sort of thumbs up, and the helix would go around the outside of the thumb to twist up, okay? And RNA is a single polynucleotide strand, right, instead of being that double helix. So DNA replication is called semi-conservative, and that's because half of the original DNA molecule is kept in each of the new molecules. So replication takes place in the S phase of interphase. So each copy, or each strand, is copied. And so what happens is the DNA is unzipped, and RNA makes a identical strand all along the DNA on each side. So in the 1950s, it was unknown how DNA replicated, and there were 3 possibilities that were suggested. There was conservative replication, where one completely new helix is made and a copy is made from the already original existing one. And then semi-conservative replication, which we know to be true today, is where each new helix contains one new and one old strand. And then the dispersive replication, where each new molecule is a whole new molecule that's made up of scattered new and old bits. Okay, so here's a model of that. So with the semi-conserved, you can see that the pink color is the newly synthesized strand, and the red is the original template. So in semi-conservative, each, the original and a copy, so the original is split up to where each new molecule has one new synthesized strand and one original template strand. And then you can see in the conservative model, the original is one complete molecule, and the newly synthesized is one complete molecule. And then in the dispersive, you can see that it's sort of this like mixed mosaic of new and original strands. So the Meselson and Stahl experiments, the Meselson and Stahl tested the hypothesis of DNA replication. So they cultured bacteria in a 15N medium. So 15N is nitrogen 15, which is an isotope to the E. coli bacteria. And the bacteria used 15 nitrogen to make their DNA, and over time, the isotope replaced all the existing nitrogen. So they did this because they knew that if the DNA is being replicated using nitrogen 15 as opposed to nitrogen 14, that the DNA would be heavier than normal nitrogen 14 DNA. So then they shifted the bacteria, which contains now the 15 nitrogen, to a 14 nitrogen medium. And the DNA was isolated at different times corresponding to the replication cycles 0, 1, and 2. After one replication cycle, the DNA was all of intermediate density. So this rules out the conservative replication model, which would predict that both heavy density DNA and light density DNA would be present, but none of intermediate density would be present. So this result is consistent with the semi-conservative replication model, which predicts that all DNA molecules will consist of one 15N and one 14N, right? It's because it's intermediate, so it's not all heavy, it's not all 15N, and it's not all light, it's not all 14N. It's intermediate between the two. The result does not rule out the dispersive replication model, which also predicts that DNA will be of an intermediate density consisting of the interspersed mosaic, right, of 15N and 14N labeled segments. But after two replication cycles, two bands of DNA were seen, one of intermediate density and one of light density. And this result is exactly what the semi-conservative model predicts. Half should be 15N, 14N, intermediate density DNA, and half should be 14N, 14N, light density DNA. So this result rules out the dispersive replication model, which predicts that after replication cycle 1, the DNA density of all DNA molecules will gradually become lower and lower, so no intermediate density DNA should remain by replication cycle 2. And so this really supported the semi-conservative model for DNA replication. And that is it. So I will see you at 16.2.